Good afternoon, everyone. And I will be telling you how we are developing novel feed ingredients from land and sea by using new technology and why this is important for a sustainable future. And Foods of Norway is a center for research-based innovation at NMBU. And it is a blue-green center that includes aquaculture, agriculture, and forestry. And we started about 3 years ago, and the center will last until year 2024. And we have a budget of $25 million. And important research that we do is to develop sustainable feed ingredients from blue and green biomass. And then we determine how these ingredients affect the performance, the growth performance, the physiology, the health, and the product quality of the fish as well as terrestrial farm animals. An important biomass that we work with is trees, spruce trees, and seaweed, as well as animal co-products. And another important research activity in the center is to develop novel or new methods that we can use in future breeding programs to develop more robust and efficient salmon that can better use these ingredients. And in Foods of Norway, we have with us 19 industry and innovation partners that helps us take the results rapidly into commercial settings, implementation of results. And this year, we also have with us BioMar and a seaweed company called Ocean Forest. And the salmon feed has changed dramatically as alternatives to fish-based feeds are being developed. And today, the salmon feed is largely consisting of plant ingredients. But the question is, how sustainable is it to use plant ingredients in the long run? And should we feed our fish food that we can eat ourselves? To secure a sustainable future, we need to think differently. And using biomass, such as trees and seaweed, becomes then an interesting alternative. We are also working with the insects, but we're still at early stages. And one of the most important sources we have in Norway is the seaweed. We have a long coastline and access to large sea areas. And this gives us opportunity to cultivate large amounts of this biomass. And another option is to use trees to make feed. And in fact, the trees is the largest biomass we have in Norway. Nearly half the country is covered by trees. And the key to unlocking these resources is the technology to convert them into microbial ingredients, such as yeast. And basically, here you see how we are actually developing feeds from the trees. And in short, you start out with a byproduct from the forest, from a tree, that is chopped into wood chips. And the tree fibers are then separated into cellulose, hemicellulose, and lignin. The cellulose, hemicellulose is converted into tree sugars. And here we're using novel enzymes that are developed at our university. The tree sugars are then put into a fermenter, and then we add nitrogen and other nutrients. And this we get from the animal and fish co-products. And then we add certain type of yeast to this bioreactor. The yeast grow, we harvest the yeast, we dry it, and that becomes a protein-rich feed ingredient that we add to the diet for the salmon, for instance. And this shows the biorefinery lab at the university. We are producing yeast in small quantities. And important things are to find the best combination of the sugars and nutrients that the yeast grow on. And the second thing is to find the best yeast strains that grow well on these substrates. And we start the fermentation in very small plastic vessels, and we gradually scale up the fermentation to larger and larger bioreactors. And last year, we scaled up the production to 200 liters. This we could do because of our industrial partners that we work closely with, Borregaard, which has the world's most advanced biorefinery and much larger fermenters that we have on campus. And this provides very important information and knowledge how producing yeast from spruce tree sugars can happen under larger scale condition, and it's an important step towards commercialization. Also, it provides us enough yeast to run experiments with salmon. Also, we recently published an article looking at the sustainability of using yeast in salmon feed. This we did together with the University of California. And yeast is a sustainable feed ingredient with a low carbon footprint. Yeast grow very rapidly. It doesn't require any agricultural land. It uses very little fresh water, and it is produced by low-value biomass that do not compete directly with human food. The potentials are great. Now, using yeast in diets for feed is not exactly new. It's been used for a long time, but more like a feed additive, whereas we are using yeast as a protein source. And when you look closer at the yeast, it has a high nutritional value. The protein content is about 50 to 60%, and it also contains a wide range of bioactive components that actually promote a good, healthy gut. And here you see the amino acid profile of the yeast, and it's very close to the requirement of the fish, with the exception that methionine level is lower than in fish meal. And we've done a series of trials with yeast in diets for salmon during different life stages. And in general, we see that when you're feeding yeast, the animals, the fish, they eat well, they grow well, and they use the feed efficiently. But the nutritional value very much depends on the yeast strain you use, how do you ferment it, and the downstream processing condition. And here you see the digestibility of different yeast-based diets compared to a high-quality fish meal. And you see there's a large differences in digestibility. On the ingredient level, you see that the fish meal diet, or the fish meal, gave a digestibility of 89%, also did Candida utilis yeast, whereas baker's yeast or Saccharomyces gave 45% digestibility, which is half of the fish meal. So basically, the downstream processing conditions is very important to increase the nutritional value of the yeast. And here we were exposing the yeast cream to different conditions, such as cell crushing at different speeds, autolysis at different times point, and spray drying at different temperatures. And we look at the electron microscope picture, you see that the autolysate didn't really affect the cell surface all that much, but cell crushing had a big impact on the cell surface. But interesting, when you look at the digestibility in salmon, we found that the yeast exposed to autolysis had the highest digestibility of about 90%. And that's because these endogenous enzymes started deteriorating the cell membrane or from the inside, and therefore it liberated the protein so that the digestibility increased. Another thing is that yeast contain a wide range of bioactive components, and it actually has a positive effect on the health of the fish. And we are testing different yeast products using cell cultures as well as intestinal cell line, whereas the best candidates we then test in the diet for salmon. And this is important because if you can document value-added effect beyond being just a protein source, it has a big impact on the profitability of the process. Our results also shows that when you feed yeast during the critical phase, such as seawater transfer, you see that the animals, the fish, the salmon, eat better, and they also have a higher growth rate. And also, based on the gene expression data, we see that these fish are more robust, and they can cope better with the stress during this time, because they have reduced inflammation in the gut, reduced oxidative stress, they have an increased gut barrier function, and also they cope better with the saltwater saltification process. So it shows that yeast is a high-quality feed ingredient with functional properties. So the question is, when can these innovations be taken into use? And you've seen the advantages, but we need to continue this research so the yeast can be a real alternative feed ingredient that can produce at large quantities and at a competitive price. And with us, we have 19 industry and innovation partners, and they will play a critical role in taking this or new feed solution into a commercial setting. With that, I thank you for your attention.
Thank you, Margaret. That's great. We've got time for a couple questions. Yeah, right behind you.
Hi. The digestibility of the yeast is significantly lower when compared to the fish meal, right? But it didn't show any difference in the growth. How does that translate it to?
Well, we saw digestibility of one yeast strain being identical to high-quality fish meal, whereas another strain actually had half the digestibility. So it depends on what strain you're using and the conditions where it's produced. I didn't show you so much growth performance data, but we see that we can obtain very similar growth performance as a high-quality fish meal. So some of the yeast strains perform very well, whereas others may not, And this is what we're looking at into now.
Okay, thank you.
Yeah, and back there?
Yeah, you showed some cell disruption and mentioned that the downstream processing would also affect digestibility. I was wondering if cell disruption improved the digestibility of some of the yeast you tested.
Well, I did show you the downstream processing, how it has a large impact on the digestibility, yes. And the most efficient processing was when the enzymes within the cell is deteriorating the cell wall, liberating the bound protein. That's what gave the best effect. But you could also, of course, add exogenous enzymes to the process, and maybe that would also open up the cell walls.
Yeah, May? Oh, okay, go ahead.
I have a question about scalability. Do you have a sense of what biomass of trees you need to produce a certain biomass of yeast at the other end?
Well, what we're doing right now is we're using the biomass from spruce trees, but you could also use different types of trees, and we're combining that with biomass from the seaweed. And we use that in the integrated biorefinery process, and then we produce yeast. And if you combine the two sugar streams, the seaweed will also provide nutrients and buffers into the solution. In that respect, you have efficient production.
With new alternative ingredients, one of the questions that comes up very often is the acceptance from end consumers. Is that something you tested at all regarding yeast inclusion in salmon feed?
Yes, it's a good question. And you can say that in Norway. I couldn't, of course, say what's happening in the US. This is a concept when you can actually use the second generation sugars, from the trees, the third generation sugars from the seaweed. You can use this renewable biomass to produce high quality fish feed that also has a good effect on animal health. It actually it has a good story, you might say. But we haven't done consumer tests. But that is also part of Foods of Norway. We would like to do that because it's so important.
So how do you make the cells autolyze? Do you have to put them through a certain physiological condition? Autolysis?
How do you actually stabilize it? Well, of course, we actually use the yeast cream, and we just maintain a stable pH, and then enzymes are active and they deteriorate the cell walls.
Thank you.
Okay, any other questions? Yeah, okay. Dave.
It sounds like your group has a pretty broad remit. How did you decide on this particular technology as the one to focus on as opposed to other ways to produce sustainable feed ingredients?
Well, I've been working with the bacteria meal from natural gas for about 20 years, which has been very interesting. Calista has now taken over the technology. And we're also looking at, of course, other microbial ingredients. And why the yeast? Well, we have to look around in Norway. We have 3% agricultural land, no way to produce crops to speak of, but we have to think about what do we have available. We have the trees, we have the seaweed, and we have the animal byproducts. And then we have the new enzymes that allow us to work with this biomass in a different way that we could ever do before. So you can now efficiently transfer these very strong fibers into sugars, and then you work on it and develop it further. So, and also yeast-based fermentation, it's easier to work with than bacteria. It's not as critical, They're pretty much robust microorganisms.
So Norway and the EU are resistant to the idea of using land animal proteins in fish feeds. What about if you're talking about microbial proteins? What if you had some sort of a microbial intermediary that would digest land animal proteins? you would then have a microbial soup that you could
True.
then use. Has that been explored, and is there
Yeah.
regulatory support for that sort of approach?
We're looking into actually using certain fungi that can use the animal byproducts, such as the fat, and convert that into a high-quality protein sources too. So it's not just yeast that we're working on. We're looking at what do we have in terms of substrates, how can we actually upgrade it by new technology, and make that a high-quality feed ingredient.
Thank you, Margaret. We really appreciate it. Very good.